Hello everyone, my name is Fakhkhari. I am a PhD student at the Faculty of Sciences, Mohammed V University of Rabat. I am delighted to share with you our lab work entitled Design of SARS CoV2 peptides recognized by the most frequent HLA alleles in the Moroccan population using immunoinformatic approach, supervised by Professor Fakhkhari. It's an in silico study aiming to identify in the short term specific SARS CoV-2 peptides of mutations isolated from strains of Moroccan patients with high affinity scores for the most frequent Moroccan HLA alleles, and in the medium term to test their effect on patient cells and controls for diagnostic purposes. So to assess the HLA peptide band and affinity predictions, we first selected SARS-CoV-2 mutations isolated from strains of Moroccan patients using GSIDE database. The table shows only 15 mutations present at the level of the protein S and sp3 and protein N that will be included in this study. Moroccan frequencies of HLA alleles were retrieved from the allele frequency net database from studies carried out on different Moroccan populations. This table shows the most Moroccan frequent alleles selected for this study for class 1 HLA-AA, HLA-B, and HLA-C, for class 2 HLA-GRB1. The HLA band and affinity predictions were run using NetMHC PAN version 4.0 for class 1, NetMHC 2 PAN version 3.2 for class 2 alleles. We predicted the peptides band and affinity of each HLA alleles to all possible overlapping 10 mer for class 1 and 50 and 14 mer for class 2 peptides. Our dataset of peptides band and affinity includes only peptides containing the mutation in their sequences with strong score of band and affinity. HLA alleles assessment is the last step to generate our results. Band and affinity frequencies of peptides were compared among the two groups, peptides of wild type and peptides of isolated mutations, to see which peptide varies its band and score of affinity in the presence of the mutation and with which allele, to identify at the end the specific peptides with high affinity scores. So, to better understand this assessment, this table shows some examples of our generated results. So, as you can see here on the table, for the mutations of these three proteins, the affinity of peptides in the presence of the mutation is very higher than peptides with no mutations. As you can see here, there is a big difference. In this study, we generated an in silico peptides of variants isolated from strains of Moroccan patients with high affinity scores for some HLA alleles to test them later on patients and controls cells. These data may provide a better understanding of the problem of host cell susceptibility and the outcome of SARS-CoV-2 infections, and may also be useful for further investigation to identify potential therapeutic targets for the vaccine strategy. And thank you for your attention.